My name is Daniel, and I'd like to share my findings on intracellular signaling differences between self-renewing and proliferating leukemia stem cells in acute myeloid leukemia. Acute myeloid leukemia, or AML, is a myeloid malignancy characterized by the expansion of abnormal immature myeloid cells. It is a lethal disease with a less than 50% two-year survival rate. Although most patients can achieve complete remission with standard therapy, many relapse and die from therapy-resistant disease. Relapse is caused by leukemia stem cells, or LSCs, which are rare cells endowed with self-renewal potential and can recapitulate the disease. Defining LSC's self-renewal mechanism will reveal therapeutic targets to prevent relapse. Evidence suggests that self-renewal and proliferation are mutually exclusive in normal hematopoietic stem cells. Our lab was motivated to see if there are LSCs with different self-renewing and proliferating potential. We use single-cell RNA sequencing to interrogate the LSC-enriched compartment of a mirror model of AML and found distinct transcriptional profiles within the LSC-enriched compartment. From single-cell gene expression profiling, we identified two cell surface markers, CD36 and CD69, that can delineate the different transcriptional signatures. We performed in vivo mouse reconstitution assays to test their self-renewal potential. We found that CD36 high LSCs are highly proliferative but have low self-renewal capacity. In contrast, CD69 high LSCs are poorly proliferative but have high self-renewal capacity. Given that the self-renewing LSCs and proliferating LSCs are transcriptionally and functionally distinct, we hypothesized that intracellular signaling is also unique for each LSC subpopulation. So we asked, what are the signaling differences between self-renewing and proliferating LSCs? Our goal is to identify dysregulated pathways that are unique to self-renewing LSCs. In order to study the signaling profiles of proliferating and self-renewing LSCs, we turn to mass cytometry, or CyTOF. CyTOF is a form of flow cytometry that can measure up to 40 proteins for thousands of cells at single-cell resolution. And our CyTOF panel includes signaling molecules that we're interested in. In this analysis, we measured the levels of the signaling molecules in 6 human AML samples and compared signaling molecules expression between self-renewing and proliferating LSCs. In this plot, each shape represents the difference of a signaling molecule expression between the two LSC populations in a single human AML sample. Red X is the average difference of each signaling molecule for the 6 human samples. Our CyTOF data revealed signaling intermediates that are significantly elevated in the self-renewing LSCs, suggesting that these signaling molecules are important for LSC self-renewal, while one-to-one comparisons reveal key signaling intermediates that are elevated in self-renewing LSCs. Such analysis cannot easily determine signaling relationships involving multiple signaling proteins. Therefore, I chose Bayesian networks to model the signaling molecule relationships. Bayesian networks is a machine learning method that surveys all possible pairwise relationships of each measured protein for intercorrelations and defines how the proteins are related in a global network. Our Bayesian network analysis reveals a highly connected graph, where each box represents a measured protein and each arrow represents influence between signaling molecules. Notably, PP30A and PNF-kappa B are upstream of many other molecules, suggesting that the levels of PP30A and PNF-kappa B determine the activity of the downstream molecules. In contrast, the level of beta-catenin are largely dependent on most other molecules without influencing them. While this model shows the overall relationship in LSCs, this model alone does not show unique signaling relationships that might exist in each LSC subpopulation. Given a shared signaling network structure for proliferating and self-renewing LSCs, we next ask whether the signaling dynamics differed between proliferating and self-renewing LSCs. Bayesian networks can be used to estimate how strongly one signaling molecule influences the other. We use this estimation to calculate the influence strength for each LSC subpopulation and identify unique signaling dynamics for each LSC subset. Purple arrows represent signaling influences that are stronger in self-renewing LSCs. Green arrows represent signaling influences that are stronger in proliferating LSCs. Gray arrows represent signaling influences that are not significantly different. Our model showed unique signaling dynamics for each LSC subset, and proteins exert influences in a cell-type dependent manner. In self-renewing LSCs, the strong molecular influences extend from PP38 to P4EBP1, suggesting that signaling pathways involving these signaling molecules are important for self-renewal. In conclusion, self-renewing and proliferating LSCs occupy different signaling states. Our Bayesian networks modeling revealed a unique signaling signature for self-renewing LSCs. In the future, I will interrogate the roles of each signaling pathways in mediating self-renewal and relapse in LSCs. Thank you.